Hi, my name is Jessica and today we will be explaining how DNA replicates itself. DNA replication is the process by which DNA makes a copy of itself during cell division. Cells need DNA replication so that they can create new cells that the body needs, such as skin cells and stomach cells. Think of DNA replication as a copy of the manual that tells the cell how to function properly. Without it, the cells will not know how to work. Cells are like little workers that need to be told what to do, and each cell has to have its own manual to do its job correctly. DNA replication occurs in 4 simple steps. First, DNA has a double helix structure that is made up of 2 complementary bases. A goes with T and C goes with G. They kind of look like a zipper that first needs to be unzipped by enzymes known as helicases in order to be copied. Second, when the DNA is separated into 2 strands, it creates a Y shape called the replication fork. Once the 2 strands are separated, DNA gets longer. Well, not really, because since DNA is like a spiral, it just gets straightened up and it appears to be longer. Third, so you now have 2 single strands of DNA, and thus do not have complementary bases. Fourth, next an enzyme called DNA ligase joins the 2 individual strands of DNA to new complementary bases, creating a new DNA.
The enzyme called helicase unzips the DNA strand, creating a replication fork. In comes DNA polymerase, which acts as the builder. It is the one actually putting down the new DNA strand. But this enzyme has to be led by primase. Primase leads the DNA polymerase and puts down primer that is made up of RNA. But since the polymerase can only go 5' to 3', this leaves a strand that is known as the lagging strand. To solve this tricky problem in the already tricky process, RNA primase has to set a lot of extra primer, which forms fragments called Okazaki fragments. An enzyme named ligase acts as the glue that puts the gaps together, which finally creates 2 double helix DNA strands, identical to the original.
Hello everyone, my name is David Guerrero and today we'll learn how DNA replication is accurate. First, we'll need to know what are stem cells. Stem cells are different from other cells in the body in 3 ways. They can divide and renew themselves for a long time, they are unspecialized so they cannot do specific functions in the body, and they have the potential to become specialized cells such as muscle cells, blood cells, and brain cells. Now, let's learn how cells ensure that DNA replication is accurate. The cell has multiple mechanisms to ensure the accuracy of DNA replication. The first mechanism is the use of a faithful polymerase enzyme that can accurately copy long stretches of DNA. One way to look at this is when you're doing homework and you're being supervised by your parent or a teacher in order for you to do it correctly. The second mechanism will be for the polymerase to catch its own mistakes and correct them. Stem cells have an extra safeguard to preserve the accuracy of the genetic information. DNA is double-stranded. The strands split apart and serve as templates for new copies. Every time a stem cell replicates its DNA and splits into 2 cells, the cell that remains a stem cell will keep the same strand of DNA. This strand is called the mother strand or the immortal strand. In this way, the stem cell can preserve the original copy of the DNA.
Now we're going to talk about how DNA serves as its own template and the 2 functions of DNA polymerase. You may be wondering what all this means, but don't worry, I'll explain very easily. Before replication can take place, an enzyme called helicase unwinds the DNA molecule from its tightly woven form. The double-stranded DNA is opened up or unzipped to give 2 single strands of DNA that can be used as templates for replication. It basically works like the zipper on your sweater. The point where the splitting starts is known as the replication fork. It has 2 strands. One is called the leading strand, which is at the top, and the other one is called the lagging strand, which is at the bottom. These unzipped sections can now be used as templates to create 2 complementary DNA strands. Each DNA molecule has 1 original strand of DNA and 1 strand made from a copy. The original strand always acts as a template for the replication. Each molecule will always have an original strand to work from. Now let's talk about DNA polymerase. DNA polymerase bonds the nucleotides together and forms new strands to match the template. For the leading strand, DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. DNA polymerase can also detect the error of a wrong nucleotide added to a strand of DNA and fix it. They remove nucleotides from the end of a strand when they discover a mismatched base.
So there you have it. I hope our team made it really easy for you to understand what DNA replication is all about.